Today, next plant growth regulator, that is cytokinins. So, cytokinins are the phytohormones which are helps in cell division. So, helps in cell division, mainly cytokinesis. Cytokinesis is nothing but cytoplasmic division. Cytoplasmic division mainly controlled by cytokinins. So, two scientists, they have discovered this cytokinins, Scoop and Miller. Scoop and Miller, they works on to discover these auxins and isolate. One thing is the internodal region of cells. So, internodal region of means the cells of internodal region of cells in stem of tobacco plant. It means internodal region of cells in tobacco stem, they show proliferation. Proliferation is nothing but division, shows division. It means the tobacco stem of internodal cells shows proliferation. Proliferation is nothing but shows division, dividing. When this shows a proliferation? With the help of auxins. It means cytokinins, they are not able to work independently. So, cytokinins are works together with another phytohormone. In this situation, auxins added with extract of what is that, vascular tissue, extraction of vascular tissue or extraction of coconut milk or extraction of yeast, extraction of DNA. These. So, cytokinins are works along with another phytohormone. Cytokinins, they cannot able to work independently. So, in these compounds, auxins are combines with these extractions of vascular tissue, coconut milk, yeast and DNA. In these four types of extractions, they have different types of compound. That chemical compound which is kinetin, that kinetin added with auxins, then after this internodal region of tobacco stem cells, they shows dividing. It means the tobacco plant stem cells are dividing by the help of auxins, and these auxins added with these extracts. These extracts contain, that is the cytokinin. So, in these extracts contain cytokinins added with auxins, then after only tobacco plant shows proliferation or dividing, cell division. So, scientist persons, first of all, they know, they are discovered and isolated these cytokinins in herring-sperm DNA. In herring-sperm DNA, Skoog and Miller, they discovered these cytokinins and given the name to them, that is kinetin. Later, we are understand this kinetins are very important for cytokinesis. Kinetins are very important for cytokinesis. That's why we are gave the name that is cytokinesis. What is that? Cytokinins. These are helps in cytokinesis of cell division, that is cytoplasmic division. In case auxins, what is that? Cytokinins and auxins both are joined together to perform a function to show a cell division. So the combination of phytohormone and auxin, what is cytokinin. So auxins and equals to cytokinin concentration. Cytokinins concentration. Result of only cell division. Only cell division. It means cytokinins works with another phytohormone. In this situation, we are taken auxins. Auxins is equal to cytokinins. It means concentration of the auxin and concentration of the cytokinins are equal. Equal in the concentration we will take when we are observed only cell division, not differentiation. But not differentiation. Only shows cell division. This combination when we take to callus formation in tissue culture. Callus formation in tissue culture. In tissue culture we are taking equal concentration of auxins, equal concentration of cytokinins. In case cytokinins are greater than auxins, shows root formation. Root formation is taking place. So when we take cytokinins are more, auxins are less in the media, this leads to root formation. In case auxins are greater than cytokinins, auxins are greater than, it means auxins concentration is more, cytokinin concentration is less. In this situation, what will happen? Shoot formation is taking place. This is called differentiation. Differentiation means differentiation of formation of roots, formation of stems, formation of remaining body parts of the plant body. That is differentiation. So when we take auxins and cytokinins equal in concentration, we only get cell division, not differentiation. When we take cytokinins are the more, auxins are less. In media, we will observe only root formation. When we take auxins are more, cytokinins are less in the media, we will get only shoot formation. This is differentiation. Next, functions. Functions of cytokinins. So first function is promote cell division. So cytokinins are mainly helps in promote cell division and cell enlargement. So second one, cell enlargement and cell differentiation. It means cytokinins are helps in mainly for cell division and cell size enlargement and cell differentiation. It is nothing but shoot formation and root formation. Next, third function is promotes adventitious roots. promotes adventitious roots. Adventitious roots. So cytokinins are promote adventitious roots in plants. It means formation of the root from any part of the plant body but except radicle. So it is adventitious roots is nothing but formation of roots from any part of plant body except radicle. Actually roots are formed from radicle. Roots are formed from radicle, but adventitious roots are formed from any part of the plant body except radicle. So this is called adventitious roots. This adventitious roots formation mainly depends on cytokinins concentration. Cytokinins are helps to adventitious roots formation. Next, fourth, leaf formation. Cytokinins are helps to leaf formation and leaf enlargement. Leaf enlargement. Cytokinins are helps to leaf enlargement and leaf formation. That is next. Fifth function, breakdown seed dormancy. Breakdown seed dormancy. It means some seeds they cannot able to germinate immediately because behind the region so many regions are there. For example, embryo is fully not developed and seed coat is not completely covered. So whatever it may be, when we seeds are not germinate, when we supply to favorable conditions, all favorable conditions. So seed is not germinate when suitable conditions also. It means seed cannot germinate at all favorable conditions also. That is seed dormancy. It means sufficient sunlight, sufficient temperature, sufficient water, sufficient hormones or sufficient anything, whatever it may be, these are all supplied to plant body, but they are supplied to seed, but seed is they cannot able to germinate. That period is there. That period is called seed dormancy period. In this seed dormancy period, when we supply all favorable condition to the seed, but seed is they cannot able to germinate. This is seed dormancy. This seed dormancy break down and to germinate, helps in germination of seed by the help of cytokinins. It means cytokinins helps to, what is that, breakdown of seed dormancy. Seed dormancy in, it means improve metabolic activities. Metabolic activities. So the cytokinins enters into that seed, the dormancy seed, and promote more amount of metabolic activity, speed of metabolic activities, then after to break down this dormancy and to helps to germination of seed. Cytokinins helps to germination of the seed to break down of the dormancy period. Next, delay of senescence. Delay of senescence. Senescence is nothing but falling of leaves. Is nothing but falling of leaves. Falling of leaves and fruit also. Falling of leaves and fruits is called senescence. So scientists, they are performed a activity. This is the leaf, this is the leaf. So this is this leaf generally dipped in cytokinins. This leaf is dipped in cytokinins. This leaf is taken as a normal leaf. This leaf, after some period also, it is evergreen. This is evergreen. It means green in color, always green in color. This is turns into yellow, turns into pale color or yellow. Why? Because yellow color leaves only, they are falling in plant body, not green color leaves. So anything yellow color leaves only will falling, we are observed in plant body. Yellow color leaves only will fall down, but green color leaves, they cannot, they are not falling. Why? Because in green color leaves, they are evergreen. They have the particular enzymes. It means we are particular cytokinins we are dipped in here. This leaf we are dipped in cytokinins. This cytokinin helps to prevent from falling of leaf. This leaf easily to fall because this leaf we are not dipped in cytokinins. When we are adding cytokinins to the leaf is becomes to evergreen, do not falling. Next, last function, formation of interfascicular interface. What is that? Promotes flowering. Promotes flowering in some plants. Cytokinins promotes what is that? Cytokinins promotes flowering in some plants. Next, formation of, next function, formation of interfascicular cambium in dicot stem during secondary growth. During the secondary growth, in secondary growth, formation of secondary xylem and secondary phloem. Outer side secondary phloem and inner side secondary xylem, which is formed by cytokinins. That is, interfascicular cambium is formed in the vascular bundle. In vascular bundle, interfascicular what is that? Cambium is formed by the cytokinins. This is helps in formation of secondary xylem and secondary phloem. Secondary phloem outer side, secondary xylem inner side to formation. This is called secondary growth. In secondary growth, this cytokinins helps to formation of interfascicular cambium. This is about cytokinins. These are the different types of functions about cytokinins. One is promote cell division and cell enlargement. Second one, cell differentiation. Third one, to formation of adventitious roots and leaf formation and leaf enlargement. Next, breakdown of seed dormancy in some seeds and delay of senescence, and promotes flowering in some plants and formation of interfascicular cambium in dicot stems in secondary growth. In next video, we are discussing about gibberellins is a next plant growth regulators. If you like this video, please like, share, comment, and for more videos, subscribe our channel. Thank you.